So good morning, everyone, also on my side, and thank you very much for joining this webinar about pluripotent stem cells culture and expansion from research to clinical applications. So today, me, Viola Stella Palladino, and my colleague, Chao Sheng, will introduce you to Miltenyi Biotec latest solutions for culture of pluripotent stem cells. So we will see first solution for classical manual expansion and handling of these cells. And then we will introduce you to our instruments for automated and GMP compliant PSC manufacturing. So in the last decade, the implementation of new type of cellular models has opened a whole new world of possibilities, both for research but also for clinical applications. In fact, induced pluripotent stem cells, also known as iPS cells or iPSCs, can be generated directly from an already differentiated somatic cells. Then they can be reprogrammed into a pluripotent state and can be further differentiated into any cell type of the human body. This, of course, overcomes all the limitation of working with cell types that would be otherwise extremely difficult to obtain by classical means. In the last decades, the pluripotent stem cell field has seen enormous advances, and these cells find now important applications in the study of the etiology of diseases, in cell replacement therapies, as well as cellular models for drug discovery and toxicity screening. But independently from what is the final application of these cells, when working with pluripotent stem cells, it's extremely important to have an established workflow for their culture in an undifferentiated state. As we will see in a few moments, a key factor to ensure the high quality of the line is the media that we use to culture our cells. So for this reason, today we will focus on culturing media that can be used for culture of PSCs under xeno and feeder-free conditions. Why is it so important? Well, this is because pluripotent stem cells are very sensitive cells, and incorrect handling or using unfitting reagents can lead to the loss of their pluripotent characteristic. And this is a major concern because just high quality PSC lines are then suitable for experimental applications. So it's clear that having a robust and stable culture environment is a fundamental requisite for successful culture and therefore successful experiments. Having this concept well in mind, we have developed a new generation of cell culture media for PSC maintenance and expansion, StemMACS PSC Brew XF. StemMACS PSC Brew supports, in fact, consistent cell growth without compromising the maintenance of stem cell specific characteristics. But which are these characteristics that we should monitor for? First of all, a good media allows PSCs to maintain their typical morphology. And here you can see that in these pictures of pluripotent stem cells that have been cultured with PSC Brew. So as you may see, cells cultured with our media maintain all the key morphological characteristic of pluripotent stem cells. Moreover, they also display classical and consistently stable doubling times over consecutive passages. A second important criteria that we should always keep in mind is that a robust media must support and allow high expression, high and actually persistent expression of pluripotency markers, also when PSC are maintained in culture for a longer time. And here you can see the assessment of this feature by flow cytometric analysis of pluripotency associated markers at different passages. As you can see from the graphs, the lines display high and persistent expression of TRA160, CA4, OCT3-4, SOX2, and these are all markers that are associated with a pluripotent state. And on the other hand, they showed always a very low expression of CA1, that is an early differentiation marker. At the same time, the media should also allow the cells to maintain their full pluripotency, also on a functional level. And the media should not introduce any bias in the differentiation capabilities of our lines. So we assess this feature by testing if PSC lines cultured in StemMACS PSC brew can differentiate into cell types that derive from the 3 embryonic germ layers. As you might already know, the 3 embryonic germ layers, so endoderm, ectoderm, and mesoderm, form really early during the development, and then with time, they will give rise to all the cells that compose the human body. So to assess if our pluripotent stem cells maintained in this condition would maintain also these pluripotent characteristics, we used a specific kit, the StemMACS 3 lineage differentiation kit. And this kit allows to induce the cells to differentiate into the 3 germ layers if they are actually pluripotent. Then we analyzed the results by flow cytometry that confirmed the ability of these cells to differentiate into mesodermal lineage, into definitive endoderm cells, and into neuroectodermal cells. Finally, we know that PSCs tend to acquire genetic aberration if maintained in prolonged culture, and this is true especially if they are mishandled. The genomic stability of the cells maintained in our media was analyzed by karyotyping and also more in detail by evaluating the burden of copy number variants present. And in both cases, we see that the karyotype was stable also after long-term cultivation, so cultivation for more than 10 consecutive passages. Okay, so after talking about important features of a culture media for the cells, let's add also something that is important for the researchers. So as just mentioned, PSC culture have distinctive metabolic demands and do not tolerate stress conditions. And this is the reason why the media changes have been traditionally performed with 24-hour interval. But by using new generation cell culture media, such as StemMACS PSC brew, more flexible feeding schedules can be applied. So you can either choose the classical everyday feeding, so every day changing the media, or you can opt for every other day feeding, or even nicer, you can skip feeding on the weekends. How did we achieve this flexibility in feeding schedules? Well, it all comes down to the ingredients that were used for this media. So for example, in the new formulation, we have a stable FGF2. This is an improved sequence FGF2 that has a higher thermostability as well as a higher resistance to proteases, while it maintains the same biological properties as a natural FGF2. And this, of course, ensures that FGF2 is present in the culture in all the feeding schedules that we want to apply. Additionally, we also have carefully improved the buffering capacity of this media. And as you can see, the StemMACS PSC brew ensures pH values within acceptable range in every feeding schedule applied. We have also evaluated the level of nutrients in all the strategies. So as you know, carbohydrates in the form of sugar, so for example, glucose, are one of the major sources of for energy for the cells in culture. And as you can see, the levels of glucose decrease physiological importantly, because the cells are, of course, using it, but we do not see differences due to the different feeding schedules that we apply. Similarly, the levels of glutamine, that it's, of course, an essential amino acid that it's also required for the proliferation of the cells. So the levels don't really show any significant difference due to the feeding schedule. Finally, we have evaluated the presence of byproducts in the culture. So as you might know, lactate accumulation in the media occurs naturally, but when we have very high levels, this might reduce the cell growth and productivity. And similarly, in aqueous solutions, such as the culture media, the glutamine will spontaneously decompose into ammonia. And also ammonia has been reported to be toxic and inhibitory for mammalian cells. So we have evaluated the concentration of these two byproducts in all different strategies, and we report that there is no significant difference due to the strategy itself. So this concept further strengthened the fact that the media can be used in a very flexible way to fit the need also of the researcher. One last feature of this new culture media is that, of course, we know that the field is evolving and that in the future, more and more application will take advantage of pluripotent stem cell line. And we want to enable the researchers to be able also to perform these more challenging applications. So, for example, gene editing or cell sorting. Therefore, we have developed a special supplement, the StemMACS PSC support, that is fully compatible with our culture media. And here you may see how these two media have been combined to support the growth of TRA160 CA4 positive cells that have been sorted using our MaxQuant title and then plated in low density in a 96 well plate. So after 10 days of culture, 6 clones were chosen and further expanded. We then perform, of course, quality control of these cells to see if they maintained all the pluripotency characteristics that we have seen before, and all the cell lines shown to be highly pluripotent and could be used for further differentiation into different cell types. So in this first part of the presentation, we saw how choosing a good culture media for pluripotent stem cells is a key point in successfully culturing and maintaining these cells. Let's now see what solution we could apply if we want to move one step further and manufacture PSCs in an automated and closed way. And for this, I leave the stage to my colleague, Ciao.
Okay, thanks a lot, Viola. Really welcome to our webinar from my side. And I'm really happy to really share our latest experience and our workflows with you. More this time on the manufacturing of PSCs, more for the clinical applications. So as I think we all know that really right now it's a very exciting time for pluripotent stem cells, as we see many scientists are already using trying to translate their research experience and their expertise into clinical applications to really address many devastating diseases. So today, I will very quickly show you some our established workflow at Miltenyi Biotec for automated, closed, and GMP-compliant manufacturing of human pluripotent stem cells that might be, very helpful and supportive for your clinical translations. Well, speaking of manufacturing of pluripotent stem cells, PSCs, and their derivatives for these clinical applications, so we all know that these are very challenging and very complex endeavors. And most of these current manufacturing processes involve multiple expensive instruments, very skilled operators, and also high-class clean rooms. Meanwhile, standardized in-process control and quality control assays are also required throughout the entire process. So, and also in some cases, like the users also wants to somehow isolate isolate the desired cell populations for downstream applications that will also require a sterile, gentle, and GMP-compliant isolation technique. So all these factors together could really limit the manufacturing skill, compromise the reproducibility, while at the same time also significantly drive up the cost. So, I mean, everyone in this field is facing the same question, the same challenge. That is, how to make these complex processes more standardized, more scalable, also at the same time more affordable for the end users. And here are some solutions from Miltenyi Biotec. So with the CliniMACS Prodigy cell manufacturing platform and all these separated open handling steps, multiple devices, the skilled operators, the high-class clean rooms, they can all be integrated into one automated and closed system. And the users can also build customized cell manufacturing workflows, including for the PSC expansion differentiation using the CliniMACS Prodigy. And then with the MaxQuant analyzer, so then standardized flow cytometry IPCQC assays can be realized with the automated technologies. And finally, with the MaxQuant title cell sorter, so this can be applied to isolate the desired cell populations with a very gentle, fast, and also GMP compliant cell sorting technology. So here today, due to the time limit, so we will focus on the CliniMACS Prodigy. So together with the single-use tubing sets, the CliniMACS Prodigy allows automated cell processing from starting materials to final products. And upon your request, so the Prodigy can actually automate all these different cell handling steps, including sample preparation, dense degraded centrifugation, cell washing, medium change, etc., up until the final formulation step. And actually, the CliniMACS Prodigy platform is very open and very flexible, and we have already developed a very broad range of software applications for various cell types and various workflows. For example, for the CAR T cell manufacturing processes. Well, for the adherent cell types, like pluripotent stem cells and their derived somatic cell types, now we have this adherent cell culture process to address these requirements. So the CliniMACS Prodigy adherent cell culture process actually allows cell cultivation to take place in two different units, the centrical unit and the external culture vessels. So first we have the centrical unit, and there actually there's a CliniMACS Prodigy chamber lies inside this unit, where you can support you can do adherent and non-adherent cell cultures. You could also automate the cell feeding, cell harvesting within the centrical unit, and you can control, of course, the temperature, the concentrations of CO2 and air. There are also a camera integrated under the chamber so that you could visualize your cell manufacturing, your cell cultivation, and there are also multiple input port available so that you can supply the cell cultures with different medium and cytokines. And this chamber actually offers a 100 square centimeter surface space. But we know that for PSC expansion differentiation, very common that you would need a larger surface space. So for this, you know, external culture vessels can also be easily connected to the CliniMACS Prodigy tubing set via stereo welding, so to enlarge the cell culture space. So for example, these multi-layer cell stacks can be very easily connected to the tubing set. And right now we have tested that you can actually connect maximally 3 of these 5-layer cell stacks at the same time to the tubing set. And with that, you will have roughly 10,000 square centimeter surface space. So it will be fairly large for the cell culture. And then with the when we designed this adherent cell culture software, actually our goal is really to make sure that it's very flexible, it's flexible enough to culture different cell types. That's why we actually modularize the individual cell handling steps. For example, now on the main menu, you can find these different modules for density gradient centrifugation, for coating, for inoculation, medium change, harvesting, etc. And the users can very freely select and combine these different modules. And as a result, so very complex workflows like direct differentiation of PSCs can be realized, can be adapted into the closed and automated CliniMACS Prodigy adherent cell culture process. And within each module, the users can also set up all the parameters according to their established SOPs. For example, in the coding module, the users can set up the coding temperature, the coding time, the volume of the coding reagents, etc., all according to their established procedures. So we have already established some workflows for GMP-compliant expansion of human MSCs and PSCs. We also have established workflows for directed differentiation of PSCs into brain dopaminergic progenitors and also cardiomyocytes on the Prodigy. So today we will focus on the GMP-compliant expansion of PSCs on the Prodigy. So for this, we have first developed the iPSC Brew GMP medium, which is manufactured and tested under the quality management system ISO 1345, and is actually also designed following the recommendations of USP 1043 on ancillary materials. So it is currently available in bottles, but very soon, then we will also offer this GMP compliant medium in bag format, and that will be a great match with the CliniMACS Prodigy for closed PSC expansion. And in our experiment, as you can see, we showed in this result, so this GMP medium enables very stable and very efficient human PSC expansion as both clusters and also as single cells. And this medium also supports very fast culture reinitiation after the cryopreservation. So this is a overview of the GMP compliant PSC expansion on the CliniMACS Prodigy using the iPSC Brew GMP medium. So we have a dedicated application sheet. So here I will not go into details, but as you can see already on this, so here you can apply different modules, for coating, for inoculation of PSCs, for culture, for medium change, and as a result, so all these steps can be automated within the CliniMACS Prodigy adherent cell culture process. And we have also performed side-by-side comparisons between manual and Prodigy-based PSC expansion using our Reinfinity antibodies and also using the StemX trilineage differentiation kit. And looking at the expression levels of these potency markers and also the trilineage differentiation potentials, and we can see that the Prodigy versus manual control, they really gave rise to very comparable results. So by using the centric unit and the different sizes of the external culture vessels, the users can actually very easily reach different PSC expansion skills using the CliniMACS Prodigy and with much less open handling steps in comparison to the manual process. We know that establishing GMP-compliant PSC banks is a very important step in many clinical applications. And for this purpose, the closed and automated CliniMACS Prodigy adherent cell culture process, together with the iPSC Brew GMP medium and our standard QC reagent, so they could be very valuable assets for the GMP-compliant PSC expansion and banking purposes. So here, just a very quick example. Very recently, Dr. Raphael Kroll from Saira Foundation in Japan has successfully actually transferred their manual clinical grade iPSC expansion workflow into the CliniMACS Prodigy adherent cell culture process. And he has presented his great work at the IACR Tokyo Symposium and also at a joint webinar with us last year. So actually, that's already everything from my side as well. Thanks a lot.